Good evening, and welcome to Dreamy Academy. Tonight, we begin a journey not across distant stars or through the deep strata of Earth, but inward, into the most intricate and astonishing landscape known to us, the nervous system. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Right now, as you listen to my voice, a universe of electrical impulses is dancing within your skull, creating the very experience of hearing, understanding, and perhaps beginning to drift. Every thought you think, every memory you hold, every flicker of emotion, all of it arises from a silent, intricate ballet of cells, exchanging whispers of information at speeds that defy our conscious perception. This is not merely a mechanism, it is the very instrument through which the universe comes to know itself. It is the most exquisite tapestry woven from matter and energy, capable of contemplating its own existence, of dreaming, of feeling the profound beauty of a quiet night. To contemplate the nervous system is to gaze upon the most intimate universal, where the laws of physics and chemistry converge to create something we call mind. It is a subject worthy of these quiet hours, rewarding patience with an understanding that deepens our sense of wonder for the sheer elegance of life. Have you ever considered what it truly means to feel? Not just a conscious emotion, but the most fundamental sense of being present, of encountering the world. Before there were eyes to see, or ears to hear, or even a brain to ponder, there was simply a response, a quiet stirring in the primordial soup, a subtle shift in the dance of molecules. This is where our journey begins, in the deep past, long before the first neuron ever fired, reaching back to the very dawn of life's responsiveness. Let's begin by considering the simplest forms of life we know, a single-celled organism, perhaps an amoeba, drifting in an ancient sea. It has no brain, no nerves, no organs. Yet it is not merely a passive blob. It moves, it seeks food, it avoids harm. How does it accomplish this? It senses. It reads the subtle language of its environment, the concentration gradients of chemicals in the water, the gentle nudge of light. Its entire surface, its delicate outer membrane, acts as a living sensor. Tiny protein molecules embedded in this boundary act like molecular antennae, exquisitely tuned to specific signals. When a molecule of sugar, for instance, brushes against one of these antennae, it causes a cascade of events within the cell, a silent conversation of proteins that ultimately guides the amoeba towards its meal. This fundamental property, the ability of living matter to react to its surroundings, is called irritability. It is the most ancient and essential precursor to what we now understand as a nervous system, a whisper of awareness in the vast silent stretches of deep time. As life grew more complex, evolving from single cells into multicellular colonies, a new challenge emerged. How would these individual cells, now bound together, communicate with one another to act as a unified whole? Imagine a colony of cells, each capable of sensing its immediate surroundings, but without a way to share that information across the entire organism. It would be like a city where every citizen knows only what happens on their own street corner, To coordinate, to move together, to hunt effectively, or to escape a threat, these cells needed a faster, more integrated system of communication. Initially, this might have been through simple chemical messengers, diffusing slowly between adjacent cells, a gentle communal murmur. But the vastness of even a small multicellular body demanded something more immediate, more precise. It called for dedicated pathways, for a specialized language of rapid signals. Picture, if you will, the first true multicellular organisms, perhaps resembling a simple sea anemone or a jellyfish, gently pulsing in the ancient waters. Their bodies are not merely collections of cells, but organized structures with distinct functions. Here we see the first tentative steps towards a nervous system. Specialized cells, elongated and delicate, beginning to form a loose, interconnected web beneath the surface of the organism. These are the protoneurons, the ancestors of the nerve cells that now form your thoughts. They are not yet gathered into a central brain, but spread throughout the organism in what we call a nerve net. Each cell in this net can sense a stimulus, a touch, a chemical change, a flicker of light, and crucially it can transmit that signal to its neighbours. The signal ripples outward, like a stone dropped into a still pond, creating a wave of information that can reach all parts of the organism. This is a beautiful and profound innovation. When a simple hydra, a tiny freshwater polyp, is touched on one side, that local stimulus doesn't just make a single cell react. Instead, the signal spreads through its nerve net, causing the entire organism to contract, pulling itself away from the perceived threat. This coordinated movement, this holistic response, is the direct result of that diffuse network of communicating cells. The signals themselves are fascinating. They are not merely chemical, but electrical. Tiny channels like molecular gates open and close in the membrane of these protoneurons, allowing charged particles, ions, to flow in and out. This creates a fleeting electrical pulse, a miniature current that travels along the cell's membrane. When this pulse reaches the end of one protoneuron, it triggers the release of chemical messengers that bridge the tiny gap to the next cell, continuing the signal's journey. This is the fundamental mechanism of nerve communication, a dance of ions and molecules that has been refined over billions of years. The elegance of this early nerve net lies in its simplicity and its distributed nature. There is no command center, no single point of failure. Every part of the net can initiate a signal, and every part can respond. It allows for basic, but effective behaviors: feeding, movement, and defense. It represents the universe learning to coordinate its own parts, to create a system where local events can have global consequences for the organism. This foundational step, the emergence of a dedicated communication system, transformed the possibilities of life. It allowed organisms to become more than just colonies. It allowed them to become individuals, capable of integrated action and a more nuanced engagement with their world. Looking back across those millions of years, we see not chaos, but structure emerging, layer upon delicate layer. The humble nerve net, spread like a fine lace curtain beneath the skin of an ancient creature, was the first blueprint for all the complexity that would follow. It was the initial answer to a fundamental question: how can a collection of cells act as one? And in its elegant solution, we find the genesis of our own awareness. So we've seen how life first learned to sense and respond, from the single cell reading chemical whispers to the simple multicellular creature coordinating its movements through a diffuse nerve net. But what happens when the demands of the environment grow more complex? When an organism needs not just to react, but to interpret, to remember, to anticipate? How does a simple net evolve into a system capable of creating the intricate tapestry of thought and feeling? That question leads us to the next profound step in this journey. The concentration of these diffuse networks, the gathering of these whispers into a chorus, and the slow, patient unfolding of the first true brain. Our last journey together left us pondering a profound question, didn't it? We marvelled at the exquisite simplicity of the nerve net, that diffuse silent ballet of cells allowing an organism like a jellyfish to respond to its world. Yet, we wondered, how does this widespread whisper of awareness coalesce into something more, something centralized, something capable of not just reacting, but interpreting, remembering, perhaps even anticipating? How does the vast oceanic expanse of the nerve net begin to fold inward, to gather itself, to form the very first flicker of what we might call a mind? Tonight, dear friends, let us continue our peaceful exploration, tracing the delicate threads of evolution that weave this incredible story. Imagine, if you will, life's grand experiment unfolding over eons. For countless millennia, creatures drifted or clung, or simply pulsed with the rhythm of the tides, their nerve nets serving them well enough. But then, a new imperative began to stir within the ancient waters, the urge for directed movement, for purposeful journeying. Consider the simple act of moving forward. If an organism is to navigate its environment with intention, to seek out sustenance, to flee from danger, to find a mate, it needs a clear sense of quote 1 and quote 2 of quote 3 and quote 4. It needs to know where it's going and what it's encountering first. This fundamental shift in lifestyle gave rise to one of the most pivotal innovations in biological architecture: bilateral symmetry. Suddenly, the organism was no longer a radially symmetrical bloom, responding equally in all directions. Instead, it became a creature with a distinct leading edge, an anterior end that would invariably meet the world head-on. Imagine a flatworm, perhaps, gliding across the seabed. Its journey is no longer a random drift. It has a direction, a purpose. And with this new purposeful movement, a remarkable consequence began to unfold. Cephalization. Cephalization, my friends, is a word that holds within it the promise of thought, of intention. It simply means the concentration of nervous tissue, sensory organs, and the mouth at this newly defined anterior, or head, end of the organism. Think of it. If you are always moving forward, the part of your body that arrives first is the part that needs to gather information most urgently. It is the part that will taste the water, feel the currents, sense the presence of light or shadow, and indeed encounter potential food or foe. It makes perfect sense, doesn't it, to place the primary senses and the command center right there at the vanguard of the journey. And so nature, ever the brilliant engineer, began to gather the scattered threads of the nerve net. Those protoneurons, once spread diffusely, started to clump together, particularly at this newly emphasized head region. These clusters, these tiny nascent command posts we call ganglia. A ganglion is, in essence, a knot of neuron cell bodies, a local processing center where information can be collected, sorted, and acted upon with greater efficiency than ever before. Picture a flatworm like the unassuming planaria. If you were to peer into its delicate structure, you would find, tucked neatly within its head, a pair of these cerebral ganglia. These aren't brains in the way we understand them today, with their intricate folds and vast networks, but they are undeniably the ancestors of brains. They represent the very first whisper of a centralized processing unit, a place where the myriad signals from the environment could begin to be integrated rather than merely triggering a local reflex. From these pioneering ganglia, nerve cords, like tiny delicate wires, began to extend down the length of the organism's body. In many early bilaterally symmetrical creatures, these cords formed a kind of ladder-like nervous system, a structured highway for communication that was a monumental leap beyond the diffuse nerve net. Now, a stimulus at one end of the body could be rapidly transmitted and processed at the head, allowing for a far more coordinated and sophisticated response. It's as if the orchestra, once playing a spontaneous, localized melody, now had a nascent conductor at its forefront guiding the entire ensemble. What were the profound advantages of this centralization, you might ask? Why did evolution favour such a dramatic reorganization? The benefits, dear traveller, were nothing short of revolutionary. Firstly, there was speed. Imagine a message needing to travel across a vast, undifferentiated network. It's like trying to shout instructions across a crowded stadium. Some might hear, some might not, and the message gets diluted. But with a centralized hub, information is channeled, focused, and processed with astonishing rapidity. This meant faster reaction times, a crucial advantage in a world teeming with both opportunities and threats. A quick decision to lunge for food or dart away from a predator could mean the difference between life and death. Secondly, and perhaps most wondrously, came the capacity for integration of information. The diffuse nerve net could trigger a localized contraction in response to a touch. But a centralized ganglion, receiving input from multiple sensory organs simultaneously, could begin to weave these disparate threads of sensation into a coherent tapestry. It could combine the faint chemical signature of food with the subtle shift in water pressure, or the shadow of a predator with a nearby vibration. This wasn't just reacting. This was the nascent ability to interpret context, to build a more holistic picture of the surrounding world. It was the first step towards understanding, and with this enhanced integration came the possibility of more complex behaviors. No longer were responses limited to simple, automatic reflexes. An organism with a rudimentary brain could now engage in directed movement, actively searching for resources, navigating around obstacles, or even exhibiting basic forms of social interaction. This was the birth of agency, the ability to make choices that were more nuanced than mere stimulus response. The world was no longer just happening to them. They were beginning to happen in the world. Moreover, this centralization laid the groundwork for something truly extraordinary, the very first glimmers of memory and learning. If an organism could integrate information, it could, in a very basic sense, store the outcomes of past interactions. Encounter a certain chemical in the water and find food. Remember that. Encounter another chemical and find danger. Avoid that path next time. This nascent capacity to adapt future behaviors based on past experiences was a monumental leap, marking the dawn of adaptive intelligence. As these first nervous systems centralized, so too did the sensory apparatus. It was a symbiotic evolution. The burgeoning ganglia needed richer, more precise information, and the environment offered opportunities for specialized sensors. At the cephalized end, alongside the nascent brain, we begin to see the emergence of dedicated sensory organs. Consider the simple photoreceptors, or eye spots, found in creatures like flatworms. These aren't complex eyes that form images, but they are exquisitely sensitive to light and shadow. They allow the organism to orient itself, perhaps seeking out dark, sheltered places where food might be more abundant, or avoiding the harsh glare of the sun. These eye spots feed directly into the cerebral ganglia, providing essential navigational cues. Alongside these, more refined chemoreceptors has developed, allowing for a keen sense of, quote, seven or, quote, atiam in the water, guiding the organism toward food sources or away from noxious substances, and yakanoreceptors. Sensitive to touch and pressure, became more localized and integrated, allowing for precise tactile exploration of the environment. Each of these specialized senses, like a dedicated antenna, brought a unique stream of data into this central processing unit, enriching the organism's internal model of its world. This increasing complexity and integration of information is truly breathtaking, isn't it? From the simple irritability of a single cell to the diffuse communication of a nerve net, we have now arrived at a point where information from multiple senses converges in a central hub. This is not just a collection of data, it is the beginning of synthesis, of understanding. The organism is no longer just a passive responder, it is an active interpreter, beginning to construct an internal representation of the external reality. This journey, from nerve net to ganglion, from radial symmetry to bilateral, from diffuse response to centralized command, is a testament to life's relentless drive towards greater awareness and adaptability. It shows us how the silent, intricate ballet of cells, once scattered, began to coalesce, to organize, to form the very foundations upon which our own complex consciousness would one day arise. As we conclude this chapter, I invite you to pause and reflect on this incredible transition. Feel the wonder of this evolutionary blueprint, this slow, deliberate construction of intelligence. The first flicker of a mind, however rudimentary, has ignited within these ancient creatures. And with that ignition, the stage is set for an even grander unfolding, for the refinement of these simple ganglia into structures of unimaginable complexity, paving the way for the very first true brains. What wonders await us as we continue to trace these pathways of awareness? We shall explore this further in the quietude of our next shared adventure. As we gently drift forward from the ladder-like simplicity of the flatworm's ganglia, a new horizon beckons, shimmering with the promise of even deeper complexity. You see, while those nascent command centers offered a revolutionary leap, integrating sensory input, coordinating directed movement, and even holding the first faint echoes of memory, life itself was far from static. The primordial seas continued their ceaseless churn, a grand crucible of adaptation, constantly demanding more, evolving new challenges and opportunities in every wave and current. Imagine, if you will, the world through the sensory portals of an early organism equipped with those rudimentary cerebral ganglia. It could detect light and shadow, taste chemicals, feel vibrations. It could move purposefully, navigate its immediate environment, and perhaps even learn to avoid a specific predator after a close call. This was a profound achievement indeed. But what if the predator grew faster, smarter, more cunning? What if the food sources became scarcer, requiring more intricate strategies to find and acquire? What if the environment itself became a labyrinth of ever-changing currents and hiding places? The simple ganglion, for all its elegance, had its limits. It was akin to a small, efficient village council, capable of handling local affairs with admirable dispatch. But as the Quotidianität of neurons grew, and as the demands of the skiers Baten became more diverse and specialized, a more sophisticated form of governance was needed. A single, undifferentiated cluster of cells, no matter how numerous, would eventually become overwhelmed, its processing power bottlenecked, its responses too slow or too generalized for a world that demanded specificity and speed. This, my friend, is where the silent, relentless pressure of natural selection began to sculpt something truly extraordinary. The very first glimmers of what we might call a true brain. It wasn't an overnight revolution, but rather a gradual, exquisite refinement, a slow unfurling of potential, driven by the ceaseless imperative to survive, to thrive, to reproduce. Consider the evolutionary journey that began to unfold. As organisms grew larger, more active, and more mobile, the need for rapid, coordinated action became paramount. A creature that could process information faster, make more nuanced decisions, and integrate a wider array of sensory data would invariably outcompete its simpler cousins. This selective pressure favored not just more neurons, but better organized neurons. It favored specialization. Think of it like this. Instead of every committee member trying to do everything, specific departments began to emerge. Some neurons became experts at processing visual cues, others at interpreting chemical signals, still others at coordinating complex muscle movements. And critically, there needed to be a central, overarching executive board to synthesize all this departmental information, to make strategic decisions, and to issue precise commands. This is the evolutionary crucible that forged the earliest divisions within the centralized nervous tissue, giving rise to what zoologists often refer to as the tripartite brain, the forebrain, midbrain, and hindbrain. These weren't fully formed, complex structures as we understand them today, but rather distinct regions, each beginning to specialize in particular functions, working in concert to create a more integrated and powerful command center. The hindbrain, often the first to show significant specialization, became the master of life's fundamental rhythms and movements. Picture an early fish, gracefully navigating the water, its hindbrain, encompassing structures akin to our brainstem and cerebellum, would be meticulously orchestrating its swimming movements, maintaining balance, and regulating essential functions like breathing and heart rate. It's the ancient, reliable foundation ensuring the organism's basic survival. It's the rhythmic pulse of existence, the silent conductor of the body's most vital unconscious symphony. Without it, the very act of living would cease. Moving forward, the midbrain began to emerge as a crucial relay station, a sophisticated intersection where sensory information from different modalities converged and was integrated. Imagine that early fish not just swimming, but reacting, perhaps spotting a flash of movement, sensing a change in water pressure, or hearing a distant vibration. The midbrain would be the bustling hub, taking these disparate signals, weaving them together, and initiating rapid reflexive responses. It's where the organism truly begins to interpret its immediate surroundings in a more holistic way, connecting sight with sound, touch with movement, fostering a richer, more immediate understanding of the present moment. It's the quick-witted responder, the immediate interpreter of the external world. And then, the forebrain. The forebrain. This is where the story truly begins to hint at the profound depths of awareness and cognition. In these early creatures, the forebrain was primarily driven by olfaction, the sense of smell. Think of how crucial smell is for a fish, detecting food sources, identifying mates, or sensing predators from afar. The forebrain became the sophisticated chemical detector, but it was more than just a sensor, it was a processor. It began to associate specific smells with memories, with rewards, with dangers. It started to build an internal map of its world based on chemical cues, leading to more complex foraging strategies and more intelligent avoidance behaviors. This region, the nascent forebrain, was the crucible where higher-order processing began to truly take shape. It wasn't yet planning complex futures or contemplating its own existence, but it was laying the groundwork for those capabilities. It was integrating sensory data with past experiences to predict future outcomes, however rudimentary. It was the first whisper of an internal world being constructed, a world where mere reaction began to transform into informed action. It was the dawning of a capacity for adaptive learning that transcended simple conditioning. What distinguished these early, tripartite brains from the simpler ganglia of their ancestors? It was the hierarchical organization, the specialization of function, and the interconnectedness of these distinct regions. Information wasn't just processed, it was analyzed, integrated, and synthesized across specialized modules. This allowed for a vastly expanded repertoire of behaviors, a greater capacity for learning and memory, and a much more nuanced interaction with an increasingly complex world. Consider the incredible advantages this new architecture conferred. A creature with a rudimentary brain could not only react to a predator, but it could also learn which specific sense or visual patterns indicated that predator. It could remember safe hiding spots or productive feeding grounds. It could even start to anticipate events based on its accumulated experience. This shift from simple stimulus response to context-dependent learned behavior marked a monumental leap in the evolution of awareness. The increasing efficiency of neural communication also played a vital role. As brains grew, the distance signals had to travel increased. Evolution, in its infinite ingenuity, began to refine the very pathways of information flow. While myelination, the fatty insulation that dramatically speeds up nerve impulses, would become more prevalent in later stages, even in these early brains, the sheer density of neurons and the increasing intricacy of their synaptic connections meant that information could be processed and transmitted with greater speed and precision than ever before. It was like upgrading from a dusty, winding dirt path to a multi-lane highway, allowing for a much greater volume of traffic to move with unprecedented swiftness. We see the evidence of these early brains in creatures like ancient fish. Their neural architecture, though still quite modest by mammalian standards, clearly demonstrates these fundamental divisions and specializations. They were, in essence, the first true navigators, the first strategists, the first creatures to truly begin building an internal model of their external reality, however simple. This journey, from the diffuse nerve net to the centralized ganglion, and now to the beginnings of a specialized tripartite brain, is a testament to the profound adaptability of life. It's a story of increasing sophistication, driven by the relentless dance of survival. Each step along this path is not just a biological innovation, it's a philosophical revelation, offering us glimpses into the very origins of our own cognitive abilities, the deep, ancient roots of our consciousness. As we conclude this exploration of the first true brains, I invite you to pause and reflect on the quiet miracle unfolding before us. From the simple irritability of a single cell to the complex decision-making of an early fish, we are witnessing the exquisite tapestry of life weaving the very fabric of mind. It is a slow, thoughtful adventure, one that reminds us of the profound interconnectedness of all living things and the deep, evolutionary currents that flow through the very core of our being. What wonders, I ask you, might emerge from this foundational design? What further complexities might these humble beginnings eventually give rise to? The journey, my friends, is only just beginning. We stand, you and I, at the threshold of an even grander unfolding. We've witnessed the humble whisper of a nerve net evolve into the decisive command of ganglia, and then blossom into the intricate tripartite brain, a marvel of ancient engineering that allowed creatures to swim, hunt, and navigate with the newfound grace. But what, you might wonder, could possibly emerge from this foundational design? How could this elegant structure, already so sophisticated, deepen its capacity for experience, for learning, for the very essence of being? The answer, my friend, lies in an expansion, a magnificent proliferation that would transform mere processing into profound perception, simple reaction into nuanced interaction. Imagine, if you will, the bustling marketplace of the early brain, where the hindbrain orchestrates the fundamental rhythms of life and the midbrain acts as a vigilant sentinel, routing crucial information. Now, turn your gaze to the forebrain, that nascent centre for olfaction and rudimentary learning. In the ongoing symphony of evolution, this region begins to swell, to stretch, to lay claim to an ever larger dominion within the cranial vault. This expansion wasn't a sudden burst, but a gradual, persistent blossoming, driven by the relentless pressures of a world growing ever more complex. As life ventured onto land, as creatures developed more sophisticated forms of locomotion, more varied diets, and more intricate social structures, the demand for greater computational power became paramount. The simple olfactory processing of early forebrains, while vital, was no longer sufficient. There was a need for a grander stage, a vaster canvas upon which the drama of existence could be painted with richer hues. And so, the forebrain began its extraordinary growth, giving rise to what we now recognize as the cerebral hemispheres. Picture them like two burgeoning lobes, mirroring each other, slowly, elegantly pushing outwards, claiming more and more space. This wasn't just an increase in size, it was an increase in potential, a silent promise of new frontiers in understanding. Within these expanding hemispheres, a thin, convoluted layer of neural tissue began to emerge and differentiate, the cerebral cortex. Ah, the cortex. Here, my dear explorer, we encounter one of the most sublime achievements of the nervous system. Initially, in creatures like early reptiles, this cortex was relatively smooth and thin, a simple, elegant sheet. Yet, even in this rudimentary form, it represented a profound leap. It was here that information, once merely relayed and reacted to, could begin to be integrated and interpreted with unprecedented depth. It was a new kind of processing, a more holistic synthesis of the world. Imagine it as the very first layer of a vast living tapestry, woven not with thread, but with the delicate, shimmering fibres of neurons. As evolution continued its patient work, this cortical layer didn't just expand. It began to fold in upon itself, a marvel of biological engineering. These elegant convolutions, the gyriamni, the ridges, and sulci, the valleys, are not mere aesthetic flourishes. No, they are a brilliant solution to a fundamental problem: how to pack an ever-increasing amount of neural real estate into a skull of limited size. Think of it like carefully crumpling a large map to fit it into a small pocket, yet retaining every detail, every intricate pathway. This folding dramatically increased the surface area of the cortex, allowing for an exponential increase in the number of neurons and the connections between them. This, in turn, meant more processing power, more capacity for learning, more space for the intricate dance of thought. With this expansion and folding came an exquisite specialization, a division of labor that allowed for unparalleled efficiency and depth of processing. Within the burgeoning cortex, distinct regions began to emerge, each dedicated to specific functions. We find sensory areas, precise territories where the raw data from our eyes, ears, skin, and tongue are meticulously processed, transforming light into sight, vibrations into sound, pressure into touch. Adjacent to these, motor areas develop, becoming the sophisticated command centers for voluntary movement, allowing for actions of remarkable precision and grace, from the delicate manipulation of an object to the powerful leap of a predator. But perhaps the most profound development within this expanding cortical landscape were the association areas. These vast regions, not directly tied to a single sensory input or motor output, became the grand integrators, the master weavers of the brain's tapestry. It is here that the symphony of sensory data, the sight of a rustling leaf, the sound of a distant cry, the scent of rain, is brought together, contextualized, and given meaning. The association areas allow for cross-modal integration, for complex problem solving, for planning future actions, and for the very act of forming an internal model of the world around us. They are where we begin to connect the dots, to understand relationships, to anticipate consequences. This is where memory truly begins to intertwine with perception, where past experiences inform present understanding. Concurrently, deep within this evolving forebrain, another crucial system was taking shape: the nascent limbic system. While not a single distinct structure, but rather a collection of interconnected regions, the limbic system began to imbue our experiences with colour, with urgency, with feeling. It linked memory to emotion, reward to survival. Here, the raw data of the senses could be tagged with significance. Is this food or danger? Is this friend or foe? This system became the seat of our deepest drives, our motivations, our capacity for pleasure and fear, for bonding and aversion. It added a powerful emotional dimension to the purely cognitive processing, ensuring that our thoughts were not just abstract computations, but were rooted in the imperative of survival and well-being. Imagine the world through the eyes of a creature possessing such an advanced nervous system. No longer merely reacting to stimuli, it could now interpret its environment, learn from its mistakes, and anticipate future events. Hunting strategies became more complex, involving planning and cooperation. Parental care, requiring sustained attention and emotional investment, became more pronounced. Social structures, demanding recognition of individuals and understanding of social cues, grew more elaborate. This wasn't just a brain. It was a burgeoning internal universe, capable of constructing a richer, more nuanced reality. The culmination of these evolutionary pressures and innovations is perhaps most vividly exemplified in the emergence of the mammalian brain. Here, the forebrain's expansion reaches truly astonishing proportions, particularly with the development of the neocortex, the new cortex. This six-layered structure, unique to mammals, represents the pinnacle of cortical evolution, responsible for so much of what we consider higher cognitive function: abstract thought, language, complex social cognition, and self-awareness. In mammals, the cerebral hemispheres become dominant, so vast that they begin to envelop and overshadow the older, more ancient brain regions. And to ensure seamless communication between these two magnificent halves, a thick bridge of nerve fibers develops, the corpus callosum. This colossal commissure acts as a superhighway, allowing information to flow freely and rapidly between the left and right hemispheres, enabling integrated processing, allowing us to perceive a unified world, to think with both logic and intuition, to experience the profound interplay of our inner and outer landscapes. From the humble nerve net, we have journeyed through ganglia and tripartite brains, to this extraordinary expansion of the forebrain, culminating in the intricate folds of the neocortex. Each step has been a deepening of capacity, a broadening of the canvas of experience. We see not just an increase in complexity, but an emergence of entirely new properties: the capacity for profound learning, for sophisticated memory, for flexible, adaptive behavior, and for the very genesis of our subjective reality. As we gaze upon this incredible evolutionary architecture, we cannot help but feel a profound sense of awe. This journey from simple irritability to the intricate folded landscapes of the mammalian brain is a testament to life's relentless drive to perceive, to understand, to connect. It is a story whispered across eons, etched in the very fabric of our being, inviting us to contemplate the miracle that allows us to reflect upon it all. What further wonders, you might ask, could possibly lie ahead in this ongoing narrative of the nervous system? What deeper layers of consciousness? What more intricate dances of thought await our discovery? And so, our quiet journey through the evolving nervous system brings us to a threshold, a precipice from which we gaze upon what is perhaps the most astonishing, intricate, and profoundly mysterious creation of all, the human brain. We have witnessed the slow, deliberate dance of evolution, from the simplest whisper of irritability in an ancient cell to the magnificent folded tapestry of the mammalian neocortex. Now, we stand at the very edge of this grand design, ready to explore the unique symphony that plays within our own skulls. What further complexities, what deeper layers of consciousness and thought might arise from this foundational design? We wondered at the close of our last chapter. The answer, as we shall see, is nothing short of breathtaking. It is a story of exponential growth, of unparalleled connectivity, and of the emergence of capacities that allow us not only to perceive the world, but to question our place within it, to dream, to create, and to understand. Imagine, if you will, the mammalian brain we just explored, a marvel of integration, memory, and adaptive behavior. Now, envision a further refinement, an explosion of processing power and organizational prowess, particularly within those already impressive cerebral hemispheres. In our own lineage, the neocortex, that thin, wrinkled outer layer, underwent a truly extraordinary expansion. It's not just about size, though our brains are indeed larger relative to our body mass than many other mammals. It's about the sheer density of neurons, the complexity of their interconnections, and the specialized functions that blossomed within this ever-growing canvas. Consider for a moment the frontal lobes. These magnificent regions, nestled right behind your forehead, are the architects of your future, the conductors of your will. While present in other mammals, in humans, they have expanded disproportionately, becoming the seat of what we call executive functions. Think of them as the sophisticated command center, responsible for planning, decision-making, problem-solving, impulse control, and the intricate choreography of social behavior. It is here that we weigh consequences, formulate strategies, and resist immediate gratification for long-term goals. They grant us the profound ability to hold a complex thought, to manipulate abstract ideas, and to project ourselves into a future that does not yet exist. Without our frontal lobes, we would be slaves to instinct and immediate sensation, unable to navigate the nuanced world of human interaction or build the civilizations we inhabit. This expansion wasn't merely about adding more space. It was about integrating information in entirely new ways, allowing for unprecedented cognitive flexibility. But perhaps the most defining characteristic, the true leap in our evolutionary journey, is the emergence of language. Think of language not just as words, but as a complex system of symbols that allows us to externalize our internal worlds, to share thoughts, experiences and knowledge across individuals and generations. It is the very fabric of culture, the engine of collective learning. The capacity for spoken and written language is intimately tied to specific regions of our neocortex, primarily in the left hemisphere for most individuals. Areas like Broca's and Wernicke's, those simplified terms for complex networks, represent specialized zones that coordinate the production and comprehension of speech. How did this miracle unfold? Imagine early hominids, their brains already complex, facing increasingly intricate social and environmental challenges. The ability to communicate precise information, there's food over there, beware of that predator, let's cooperate on this task, offered an immense survival advantage. Over countless millennia, the neural circuits that once facilitated basic vocalizations and gestures became refined, integrated, and expanded, giving rise to the nuanced phonetics, grammar, and semantics that define human language. With language came the ability to articulate abstract concepts, to discuss not just what is, but what might be, what was, and what should be. It allowed for the development of complex narratives, the transmission of accumulated wisdom, and the very foundation of our shared human experience. This capacity for language, in turn, fueled the development of symbolic thought and abstract reasoning. We are not merely reacting to the world, we are interpreting it, modelling it, and transforming it through symbols. Mathematics, art, music, philosophy, and science itself are all expressions of this extraordinary capacity. Our brains can manipulate ideas that have no direct physical referent: justice, freedom, infinity, time. We can create internal models of reality, predict outcomes, and engage in what-if scenarios, all within the silent theatre of our minds. This ability to detach thought from immediate sensory input, to play with concepts and theories, is a hallmark of human cognition, deeply rooted in the interconnectedness of our vast neocortex. It's as if the brain, having mastered the art of processing the external world, turned its gaze inward, using its immense power to explore the landscape of its own thoughts. And through this intricate dance of specialized regions and boundless connections, we arrive at the profound question that has captivated thinkers for millennia: consciousness itself. What is it that allows you to experience the redness of a sunset, the warmth of a touch, the bittersweet pang of memory? How does the firing of billions of neurons, the silent, intricate ballet of electrical impulses and chemical messengers, give rise to the subjective feeling of being you? This is the ultimate mystery, the grand culmination of our evolutionary journey, and yet, in many ways, it remains an enigma. While we do not fully understand the precise neural correlates of consciousness, we can certainly appreciate the immense complexity required for it to emerge. It is not localized to a single region, but appears to be an emergent property of the entire system, a grand symphony played by the entire brain. It involves the integration of sensory information, memories, emotions, intentions, and our internal models of the world. It's the constant dynamic interplay between our neocortex, which processes and interprets, our limbic system, which imbues experience with emotional meaning, and our deeper brain structures, which maintain our fundamental state of being. Self-awareness, the ability to recognize oneself as a distinct entity with a past, present, and future, is a profound expression of this consciousness, deeply intertwined with our frontal lobe functions and our capacity for language. We don't just experience, we know that we experience. We don't just think, we think about our thinking. Moreover, our brains are not just wired for individual thought, they are profoundly social brains. The human brain has evolved in the context of complex social groups, and this is reflected in its architecture. We possess an innate capacity for empathy, for understanding the intentions and emotions of others, often called theory of mind, and for forming intricate social bonds. Our mirror neuron systems, for example, allow us to internally simulate the actions and feelings of others, fostering connection and learning. This social intelligence, supported by the advanced capacities of our neocortex and limbic system, is crucial for cooperation, culture, and the very survival of our species. We are, in essence, designed for connection, our brains constantly seeking patterns and meaning in the actions and expressions of those around us. So, here we are, at the very pinnacle, so far, of this extraordinary evolutionary saga, from a single-celled organism's rudimentary response to light or chemical gradients, to the diffuse nerve net of a hydra, to the centralized ganglia of a flatworm, the tripartite brain of an ancient fish, the cortex of a reptile, and the neocortex of a mammal. we have traced a path of ever-increasing complexity, integration, and adaptive capacity. Each step built upon the last, refining, expanding, and specializing, driven by the relentless pressures of survival and the endless possibilities of life. The human brain, with its vast frontal lobes, its unique capacity for language, its ability for abstract thought, and its profound, albeit mysterious capacity for consciousness, represents an unparalleled achievement of natural selection. It is a universe within a universe, a 3-pound marvel of biological engineering that allows us to ponder the very cosmos that created us. It is the instrument through which we experience joy and sorrow, fear and love, curiosity and wonder. It is the seat of our identity, our memories, our aspirations. Our exploration of the nervous system's evolution brings us to this point of awe, recognizing that the journey is far from over. The human brain, in all its magnificence, is not a static endpoint but a continuous process, shaped by experience, learning, and the ongoing dance between our genes and our environment. It is a testament to the power of slow, deliberate change, a reminder that even the most profound complexities arise from simple beginnings. As we conclude this chapter, I invite you to pause and reflect on the incredible journey we have undertaken. From the first whisper of awareness to the symphony of the human mind, each stage has been a profound step in the unfolding story of life. And though we have unveiled many layers, the ultimate mystery of how this biological machinery gives rise to our rich inner world remains a captivating frontier. The more we learn, the more profound the wonder becomes, inviting us to continue our peaceful explorations into the silent, intricate ballet that defines us. What further wonders, what deeper understandings await our patient curiosity? The journey, I assure you, continues. Having journeyed through the vast, awe-inspiring landscapes of the nervous system's evolution, from the first whisper of an amoeba's awareness to the grand symphony of the human mind, we've marvelled at the emergence of consciousness and the profound complexities of our social brain. We've seen how life, through countless eons, built ever more intricate structures to sense, process, and respond to its world. But now, my dear fellow explorer, let us pause. Let us descend from the heights of philosophical wonder and zoom in, closer than ever before, to truly understand the fundamental building blocks of this magnificent edifice. For beneath the grand orchestrations, beneath the intricate dances of thought and emotion, lies a universe of individual performers, each playing a vital role. What are these fundamental units? How do they communicate? How do they, in their silent, intricate ballet, weave together the very fabric of our being? Today, we embark on a peaceful exploration of the neuron and its indispensable companions, the glial cells. These are the foundational elements, the very language of life itself, that allow a jellyfish to pulse through the ocean and a human to compose a sonata or ponder the cosmos. Imagine, if you will, the nervous system not as a monolithic entity, but as an exquisite tapestry woven from billions of individual threads. Each thread in its own right is a marvel of biological engineering, capable of receiving, processing, and transmitting information with breathtaking speed and precision. This thread, this star of our show, is the neuron. When we speak of the nervous system, it's the neuron that often springs to mind, and for good reason. It is the primary cell type responsible for electrical signaling, the cell that carries the messages that define our experience. But what does a neuron look like, and how is it structured for such a specialized task? Let's visualize one together. At its heart lies the soma, or cell body. This is the neuron's control center, its metabolic engine room. Here, the nucleus resides, containing the genetic blueprints for life and all the cellular machinery necessary to keep the neuron alive and functioning. It's where proteins are made, where energy is generated, and where the neuron's overall health is maintained. Think of it as the central hub of a miniature city, bustling with activity, ensuring all operations run smoothly. Branching out from the soma like the delicate limbs of a miniature tree reaching towards the sky are the dendrites. These are the neuron's primary receivers, its molecular antennae. They are typically short, highly branched structures, forming an elaborate network designed to capture signals from other neurons. Each dendrite is studded with specialized receptors waiting patiently to bind to chemical messengers from neighboring cells. The sheer complexity of a neuron's dendritic tree can be astounding. A single neuron might receive input from thousands, even tens of thousands of other neurons through these intricate branches. It's here that the neuron listens, sifting through a constant chorus of incoming information. Once these signals are collected and integrated within the soma, a decision is made. If the incoming signals are strong enough, if the vote of information reaches a certain threshold, the neuron will generate its own message. This message travels down a single, long, slender projection called the axon. The axon is the neuron's transmission cable, its dedicated highway for sending information. While some axons are incredibly short, connecting nearby neurons within a tiny brain region, others can stretch for remarkable distances. Consider the axons that run from your spinal cord all the way down to the muscles in your toes, spanning several feet. At the very end of the axon, it typically branches again, forming axon terminals or synaptic knobs. These are the neuron's delivery points, its outgoing communication stations. It's here that the neuron prepares to pass its message on to the next cell in the circuit, whether that be another neuron, a muscle cell, or a gland. So, how does a neuron actually send a message? It's a breathtakingly elegant process, a rapid, self-propagating wave of electrical change known as an role action potential. Imagine a tiny controlled spark, a sudden rush of electrical energy that races down the axon. This isn't a continuous flow like electricity through a wire. Rather, it's a dramatic, fleeting reversal of electrical charge across the neuron's membrane. At rest, the inside of a neuron is slightly negatively charged compared to the outside, maintained by a careful balance of charged particles or ions, particularly sodium and potassium. When an action potential is triggered, tiny protein channels embedded in the axon's membrane snap open, allowing positively charged sodium ions to flood into the cell. This sudden influx rapidly depolarizes the membrane, making the inside temporarily positive. This electrical flip then triggers the opening of adjacent channels further down the axon, propagating the spark forward like a domino effect. Immediately afterward, other channels open to allow potassium ions to rush out, restoring the negative charge and preparing that segment of the axon for the next signal. This action potential is an all-or-none event, meaning it either fires completely or not at all, like flipping a light switch. There's no such thing as a half action potential. The strength of a signal isn't determined by the size of the action potential, but rather by the frequency with which these sparks are fired. A stronger stimulus might lead to more frequent action potentials, a rapid-fire burst of electrical pulses. It's a remarkably efficient and robust way to transmit information over distances. But what happens when this electrical spark reaches the axon terminal? This is where the magic of the synapse unfolds, the crucial junction where one neuron communicates with another. The synapse isn't a direct physical connection. There's a tiny, almost imperceptible gap between the axon terminal of the transmitting neuron, the presynaptic neuron, and the dendrite or soma of the receiving neuron, the postsynaptic neuron. This gap is called the synaptic cleft. When an action potential arrives at the axon terminal, it triggers a cascade of events. Tiny sacs called vesicles, filled with specialized chemical messengers called kernus neurotransmitters, fuse with the presynaptic membrane and release their contents into the synaptic cleft. These neurotransmitters are like molecular keys, drifting across the narrow gap to bind with specific receptor proteins on the postsynaptic neuron. The binding of these chemical keys to their molecular antennae on the postsynaptic neuron causes a new electrical change in that cell. Depending on the specific neurotransmitter and receptor, this change can either be excitatory, making the postsynaptic neuron more likely to fire its own action potential, or inhibitory, making it less likely to fire. It's a delicate balance of pushing and pulling, whispering go or stop. Consider the sheer diversity here. There are dozens of different neurotransmitters, each with its own unique message and often its own specific effects. Acetylcholine for muscle contraction and memory, dopamine for reward and motivation, serotonin for mood and sleep, GABA, the primary inhibitory neurotransmitter that calms the brain, glutamate, the primary excitatory one that drives learning and memory. This chemical language allows for incredible nuance and complexity in neural communication, shaping everything from our simplest reflexes to our most profound thoughts. Once their job is done, these neurotransmitters are quickly removed from the synaptic cleft, either broken down by enzymes or reabsorbed by the presynaptic neuron, ensuring that the signal is precise and transient, ready for the next incoming message. For a long time, we thought neurons were the sole stars of the nervous system, the only cells truly responsible for its incredible functions. But over the past few decades, our understanding has broadened immensely, revealing the unsung heroes, the crucial support staff, the silent partners in this intricate ballet, the glial cells, or simply glia. Glia, derived from the Greek word for glue, were once thought to simply hold neurons together, but we now know they are active, indispensable participants in every aspect of nervous system function. In fact, there are far more glial cells than neurons in our brains, outnumbering them by a factor of 10 or more in some regions. They are the caretakers, the managers, the vital infrastructure that allows neurons to perform their astonishing feats. What do glia do? Their roles are incredibly diverse and essential. Structural support. Yes, they do provide physical support, helping to organize and maintain the delicate architecture of the brain. Nutrient supply and waste removal. Glia act as the brain's internal chefs and janitors. They regulate the chemical environment, supplying neurons with vital nutrients, clearing away metabolic waste products and absorbing excess neurotransmitters from synapses, thereby fine-tuning neural communication. Myelin sheath formation. This is one of their most celebrated roles. Specialized glial cells, oligodendrocytes, in the central nervous system, brain and spinal cord, and Schwann cells, in the peripheral nervous system, wrap around axons like electrical tape, forming a fatty insulating layer called the myelin sheath. This myelin isn't just insulation, it dramatically increases the speed at which action potentials can travel down the axon, allowing signals to leap from one gap in the myelin to the next. Without myelin, our nervous system would be impossibly slow, and complex thought or rapid reflexes would be impossible. Imagine turning a dirt road into a superhighway. That's the power of myelin. Modulating synaptic activity. Glia are not passive observers. They actively monitor and influence synaptic communication, even forming their own synapses with neurons. They can release their own chemical signals, affecting how neurons talk to each other, playing a crucial role in learning, memory, and plasticity. Immune response and repair. Other types of glia, like microglia, act as the brain's immune cells, constantly surveying the environment, clearing debris and responding to injury or infection. They are the brain's first line of defense. Blood-brain barrier. Astrocytes. Star-shaped glial cells play a critical role in forming and maintaining the blood-brain barrier, a highly selective filter that protects the brain from harmful substances in the bloodstream while allowing necessary nutrients to pass through. So, as we reflect on the incredible journey of consciousness and the intricate workings of the human mind, let us never forget the fundamental elegance of these cellular components. Billions of neurons, each a tiny electrical powerhouse, constantly receiving, integrating, and transmitting information through a sophisticated dance of electrical impulses and chemical whispers, and surrounding them, supporting them, nurturing them, and actively participating in their every function are trillions of glial cells, the unsung heroes orchestrating the perfect conditions for the universe of electrical impulses to dance. It is in this silent, intricate ballet of cells, in the precise choreography of ions and neurotransmitters, that the very foundation of our existence is laid. It is here, at this microscopic level, that the language of life is spoken, enabling everything we perceive, feel, think, and do. We are truly an exquisite tapestry woven from these remarkable threads, constantly being rewoven with every experience, every thought, every peaceful exploration of our own wondrous inner world. And with this foundational understanding, we are now ready to explore how these individual conversations assemble into larger networks, giving rise to the astonishing functional architecture of the nervous system. As we gently drift from the astonishing ballet of individual neurons and their tireless glial companions, we find ourselves ready to zest upon an even grander spectacle, how these countless microscopic wonders organize themselves into the majestic integrated whole we call the nervous system. Imagine, if you will, moving from admiring the exquisite brushstrokes of a painting to stepping back and beholding the entire masterpiece, its composition, its interplay of light and shadow, its profound narrative. The nervous system, in its breathtaking entirety, is not a chaotic jumble of cells, but a meticulously organized hierarchical structure divided into two vast interconnected realms, the central nervous system, CNS, and the peripheral nervous system, PNS. Think of it as the ultimate command center and its vast network of ambassadors and messengers constantly in a profound, intimate dialogue. Let us first turn our attention to the central nervous system, the very heartland of our inner universe. This is where the grand decisions are made, where our thoughts take flight, where our emotions surge, and where the raw data of existence is transformed into the rich tapestry of our subjective experience. The CNS is comprised of two irreplaceable components, the brain and the spinal cord. The brain, as we've journeyed through its evolutionary ascent, truly stands as the undisputed sovereign of this realm. It is the intricate, pulsating organ nestled within the protective fortress of your skull, a universe unto itself, where billions of neurons hum with electrical life. Here, the cerebral hemispheres, with their vast, convoluted landscapes of gyri and sulci, continue their ceaseless work of perception, memory, language, and abstract thought. Below them, the cerebellum orchestrates the elegant symphony of movement and balance, allowing us to walk, dance, and reach with graceful precision. And at the very base, the brainstem, a primordial pillar, stands guard over the most fundamental rhythms of life, our breathing, our heart rate, the silent, unwavering drumbeat of our existence. The brain is the ultimate integrator, the weaver of all sensory threads into a coherent present moment, the architect of our future actions, and the quiet keeper of our past. Extending downwards from this magnificent command center, a vital conduit, a superhighway of neural information, is the spinal cord. Encased within the sturdy fortress of your vertebral column, this slender rope-like structure is far more than a mere cable. It is the indispensable bridge, the two-way thoroughfare that allows the brain to communicate with the vast expanse of the body, and for the body to relay its myriad sensations back to the brain. Sensory information, the warmth of a touch, the bite of cold, the stretch of a muscle, races up the spinal cord to the brain for interpretation. In turn, motor commands, instructing muscles to contract or relax, surge down from the brain, traversing the spinal cord to reach their destinations. But the spinal cord is also a master of independent action, a local hero in its own right. It is the seat of our reflexes, those lightning fast, involuntary responses that often occur before the brain even has time to fully register what has happened. Imagine touching something searing hot. Long before your conscious mind comprehends the danger, your hand has already recoiled. This immediate protective withdrawal is orchestrated by a reflex arc within the spinal cord, a testament to its efficiency and its role in keeping us safe. It's a beautiful example of the nervous system's layered intelligence, where immediate survival responses can bypass the higher processing centers for speed. Given the unparalleled importance and exquisite delicacy of the brain and spinal cord, nature has endowed them with an extraordinary series of protective measures. They are, after all, the most precious jewels in our biological crown. First, the hard, unyielding armour of the skull and vertebral column provides a formidable physical barrier. But beneath this bony shield lies a more subtle yet equally vital system of defence, the meninges. These are three distinct layers of protective membranes, the dura mater, arachnoid mater, and pia mater, that snugly envelop the brain and spinal cord, cushioning them and holding them gently in place. Think of them as the finest silk linings within a treasure chest, preventing any jarring or friction. And within these meningeal layers flows the cerebrospinal fluid, CSF, a clear, colourless liquid that surrounds the entire CNS. This remarkable fluid acts as a buoyant cushion, effectively reducing the brain's net weight, allowing it to float gracefully within the skull, shielded from the forces of gravity and sudden movements. It also plays a crucial role in nourishing the brain and spinal cord, carrying away metabolic waste products, maintaining a stable chemical environment, and even helping to regulate intracranial pressure. It is a silent, constantly circulating river of life, protecting and sustaining our most vital tissues. Finally, we encounter one of the most remarkable and mysterious guardians of the CNS, the blood-brain barrier. This is not a physical wall, but a highly selective filter formed by specialized endothelial cells lining the capillaries within the brain, along with their glial partners, the astrocytes. This barrier meticulously regulates which substances can pass from the bloodstream into the delicate neural tissue, preventing harmful toxins, pathogens, and even many circulating molecules from reaching the brain. It is an exquisite gatekeeper, ensuring the brain's pristine environment is maintained, a silent sentinel constantly vigilant against intruders. This level of protection underscores the profound significance of the CNS as the very core of our being, a sacred vault housing the essence of who we are. Now let us journey beyond the protective confines of the CNS and explore the vast network that extends throughout the rest of our body, the peripheral nervous system, PNS. If the CNS is the central command, the PNS is its intricate array of scouts, messengers, and action teams, extending its reach to every muscle, every organ, every sensory receptor from the tips of our toes to the follicles of our hair. It is the interface between our internal world and the external environment, the system that allows us to perceive, react, and engage with the world around us. The PNS is itself divided into two major branches, each with distinct yet intimately connected roles. First, we have the somatic nervous system. This is the realm of conscious control and voluntary action. When you decide to lift your arm, to speak, to walk, or to consciously feel the texture of a leaf, it is your somatic nervous system at work. It carries sensory information from the body's receptors in the skin, muscles, joints, to the CNS, allowing us to perceive our surroundings and our body's position in space, and it carries motor commands from the CNS to our skeletal muscles, enabling us to execute our intentions. It is our direct, willful connection to the physical world, our instrument for deliberate interaction and expression. Then there is the autonomic nervous system, ANS, the silent, unseen conductor of our internal orchestra. This system operates largely below the threshold of our conscious awareness, tirelessly regulating the involuntary functions essential for life, the steady beat of our heart, the rhythmic expansion and contraction of our lungs, the intricate processes of digestion, the precise adjustments of our blood pressure and body temperature. The ANS ensures that our internal environment, our milieu interieur, remains stable and balanced, adapting seamlessly to the ever-changing demands of our existence without us ever having to consciously direct it. It is the epitome of the body's innate wisdom, a testament to the elegant efficiency of life. The ANS, in turn, has its own fascinating subdivisions, often described as opposing yet complementary forces, constantly striving for a dynamic equilibrium. First, meet the sympathetic nervous system, often famously dubbed the fight or flight system. This is the part of you that mobilizes your body's resources in times of perceived stress, danger, or intense excitement. When faced with a sudden threat, or even just the thrill of a challenge, your sympathetic nervous system springs into action. Your heart rate accelerates, breathing quickens, blood is diverted to your muscles, pupils dilate, and non-essential functions like digestion temporarily slow down. It's an ancient, primal response, honed over millions of years of evolution, designed to prepare you for immediate action, to confront the challenge or to make a swift escape. It is the surge of adrenaline, the sharpened focus, the heightened awareness that can save your life. Then we have the parasympathetic nervous system, the calming counterpoint, affectionately known as the rest and digest system. Once the perceived threat has passed, or when you are simply in a state of tranquility and safety, the parasympathetic nervous system takes the reins. It works to conserve energy and promote restorative processes. Your heart rate slows, breathing normalizes, pupils constrict, and digestive activity resumes. It encourages relaxation, facilitates nutrient absorption, and allows the body to repair and replenish itself. It is the quiet sigh after a long day, the gentle rhythm of sleep, the profound sense of peace that allows for healing and growth. The elegant interplay between the sympathetic and parasympathetic systems is a marvel to behold. They are not simply on-off switches, but rather a delicate balance, a continuous negotiation that fine-tunes our internal state to perfectly match the demands of our external reality. They constantly adjust minute by minute, ensuring we are neither perpetually overactivated nor excessively subdued, but rather in a state of optimal readiness and restorative calm. And finally, within the vast architecture of the PNS, nestled within the walls of our digestive tract, lies a fascinating and surprisingly independent network of neurons known as the enteric nervous system, ENS. Often referred to as the second brain, the ENS contains hundreds of millions of neurons, more than in the entire spinal cord, capable of operating largely autonomously. It orchestrates the complex processes of digestion, from sensing the contents of your gut to controlling muscle contractions that move food along, and regulating the secretion of digestive enzymes. While it can function independently, it is also in constant communication with the brain, influencing our mood, stress levels, and even our cognitive functions in ways we are only just beginning to fully comprehend. This intricate gut-brain axis reveals yet another layer of the nervous system's profound interconnectedness, demonstrating that even our deepest, most visceral experiences are part of this grand neural symphony. So, you see, the nervous system is not merely a collection of isolated parts, but a magnificent integrated whole. The CNS, with its immense processing power and decision-making capacity, is constantly receiving, interpreting, and sending signals via the PNS. The PNS in turn gathers sensory intelligence from every corner of the body and every facet of the environment, relaying it back to the CNS and then faithfully executing its commands. It is a vast, intricate feedback loop, an endless conversation between our inner and outer worlds, all working in concert to create the unified, coherent experience of being you. This seamless integration allows us to perceive a vibrant sunset, feel the warmth of a loved one's hand, formulate a complex thought, and then physically act upon it. It is the underlying architecture that supports every conscious thought, every involuntary breath, every emotion that colours our perception, and every movement that defines our interaction with the world. As we consider this magnificent organisation, from the protected core of the CNS to the far-flung reach of the PNS, we are once again invited to pause in quiet awe, contemplating the sheer brilliance of life's design and the profound mystery of how such intricate machinery gives rise to the entirety of our living experience. We've journeyed through the intricate landscapes of the nervous system, from its earliest stirrings in single-celled life to the dazzling complexity of the human brain. We've explored the silent, intricate ballet of neurons and glia, the sophisticated highways of the CNS and PNS, and the delicate dance of electrical impulses and chemical messengers that orchestrate every thought, feeling, and action. And yet, as we paused at the close of our last chapter, a profound question lingered, a whispered enigma at the heart of it all. How does this magnificent biological machinery, this universe of dancing electrical impulses, give rise to the subjective, rich tapestry of our inner world, to consciousness itself? It is a question that philosophers have pondered for millennia, and scientists continue to unravel, piece by exquisite piece. While the full answer remains a horizon we perpetually approach, we can, together, explore the very first brushstrokes of that tapestry, the initial raw data from which our conscious experience is woven. For before we can think, feel, or act, we must first perceive. We must gather information from the vast, vibrant world outside and translate it into a language our brain can understand. This, my friend, is the realm of our senses, the magnificent gateways through which reality flows into the inner sanctuary of our being. Imagine for a moment that you are not simply experiencing the world, but actively building it moment by moment. Your brain is not a passive receiver, absorbing reality like a sponge. Instead, it is an active, brilliant architect, constructing an internal model of the external world based on the whispers and shouts it receives from your sensory organs. This is a crucial distinction, for the world we perceive is not merely the world as it is, but the world as our brain interprets it. How does this extraordinary construction begin? It starts with what we call sensory transduction, a marvel of biological engineering where physical energy from the environment is transformed into the electrical language of the nervous system. Think of it as a universal translator, converting light waves, sound vibrations, chemical molecules, and pressure into action potentials that can travel along nerve fibers. Consider your eyes, those luminous windows to the world. What they truly detect are photons, tiny packets of light energy. But you don't see photons. Instead, specialized cells in your retina, the photoreceptors, contain pigments that absorb these photons. This absorption triggers a cascade of biochemical reactions, ultimately leading to a change in the electrical potential across the photoreceptor cell membrane. This electrical signal is then passed on to other retinal neurons, eventually generating action potentials that travel along the optic nerve. Your brain, nestled in the darkness of your skull, never directly experiences light. It only ever receives these electrical impulses. Yet, from this intricate sequence, you perceive the dazzling spectrum of colours, the intricate dance of shadows, the profound depth of a starlit sky. Is that not an astonishing magic? The same principle applies across all our senses. Your ears, those exquisite molecular antennae, do not sound waves directly. They capture vibrations in the air, which are then funneled down the ear canal to vibrate the eardrum. These vibrations are amplified by tiny bones and then transmitted to the fluid-filled cochlea. Here, delicate hair cells, embedded within a membrane, are bent by the fluid's movement. This mechanical bending is the critical step of transduction for hearing. It opens ion channels in the hair cells, generating electrical signals that are relayed to the auditory nerve. From this symphony of vibrations, your brain constructs the melody of a bird's song, the comforting murmur of a loved one's voice, the distant rumble of thunder. Touch, too, is a testament to this transformative process. Scattered across your skin within its layers are a myriad of specialized mechanoreceptors. Some respond to gentle pressure, others to firm indentations, some to vibrations, others to stretch. When a physical force deforms these receptors, it mechanically opens ion channels in their membranes, generating a graded electrical potential. If this potential reaches a certain threshold, an action potential is fired, sending a message along sensory neurons. This is how your brain feels the warmth of a comforting hand, the cool smoothness of a polished stone, the gentle caress of a breeze. It's not the touch itself, but the electrical echo of that touch that registers within your awareness and taste and smell. These are chemical senses relying on chemoreceptors. In your nose, olfactory receptor neurons possess molecular antennae that bind to specific airborne chemical molecules. Odorants. This binding triggers a biochemical cascade, leading to the generation of electrical signals. Similarly, on your tongue, taste buds contain taste receptor cells that bind to specific dissolved chemical molecules, tastants, initiating electrical responses. From these chemical keys fitting into molecular locks, your brain conjures the rich aroma of baking bread, the sweet burst of a ripe berry, the bitter tang of coffee. Even our internal senses, often operating beneath the surface of conscious awareness, follow this same pattern. Proprioceptors in our muscles and joints tell our brain about the position and movement of our body parts, allowing for coordinated action without constant visual monitoring. Vestibular receptors in our inner ear detect head movements and gravity, providing our brain with crucial information for balance and spatial orientation. Each of these specialized cells, whether responding to light, vibration, pressure, or chemical compounds, performs the same miraculous feat, converting a specific form of environmental energy into the universal electrical language of the nervous system. Once these electrical signals are generated at the periphery, they embark on a carefully orchestrated journey towards the brain. For most sensory information, with the notable exception of smell, which takes a more direct route, these signals first travel along the peripheral nerves, through the spinal cord, and ascend to the brainstem. From there, they are typically routed through a remarkable structure deep within the brain called the thalamus. Imagine the thalamus as the grand central station of sensory information. Almost all sensory inputs, before reaching their final destination in the cerebral cortex, pass through this vital relay station. The thalamus acts as a sophisticated filter and gatekeeper, organizing and directing the incoming streams of data to the appropriate processing areas. It ensures that the visual signals go to the visual cortex, auditory signals to the auditory cortex, and so on. It's a crucial step in ensuring that the brain receives a coherent and organized flow of information. From the thalamus, these electrical messages fan out to specific regions of the cerebral cortex, the folded outer layer of the cerebrum that we explored in previous chapters. Here, in these specialized cortical areas, the true magic of perception unfolds. Take, for instance, the primary visual cortex, located at the very back of your brain in the occipital lobe. When electrical signals from your eyes, having passed through the thalamus, arrive here, they are not yet fully formed images. Instead, they represent raw features, lines, edges, contrasts, movements. Different neurons in this area are tuned to respond to specific orientations or directions of movement. It's like an army of specialized detectors, each picking up a tiny piece of the visual puzzle. Similarly, the primary auditory cortex, nestled within the temporal lobe, processes the raw elements of sound, pitch, loudness, timbre. The primary somatosensory cortex, located in the parietal lobe, receives and processes information about touch, temperature, pain, and proprioception from different parts of your body. Each of these primary sensory areas is a hub for initial fundamental processing. But perception is far more than just registering raw sensory data. It's about making sense of that data, giving it meaning, integrating it into a coherent whole. This is where the magnificent association areas of the cortex come into play. Surrounding the primary sensory areas, these regions are responsible for higher order processing. They take the basic features processed by the primary areas and combine them, compare them with stored memories, and imbue them with context and significance. For example, when you see a face, your primary visual cortex registers the lines, shapes, and colors. But it's the visual association areas that recognize it as a face, and further still, recognize it as the face of a friend. They integrate visual information with memories of that person, with the emotional associations, with knowledge of their identity. The same happens with sound. Your primary auditory cortex hears a sequence of notes, but your auditory association areas recognize it as a melody, and perhaps even as a specific song that evokes a particular memory or feeling. This active construction of reality means that what you perceive is always, to some extent, a subjective interpretation. Your brain fills in gaps, predicts outcomes, and filters out irrelevant information based on your past experiences, your current emotional state, and your expectations. Think about optical illusions, or how two people can witness the same event and recall it differently. This isn't just about faulty memory, it's about the active, interpretive nature of perception itself. Your brain is a master storyteller, weaving narratives from the raw sensory threads it receives. Furthermore, our experience of the world is rarely confined to a single sense. We see a vibrant sunset while feeling the cool evening air and smelling the distant scent of pine. This multimodal integration is another marvel of the nervous system. Different sensory areas communicate extensively with one another, allowing the brain to combine inputs from various senses to create a richer, more comprehensive understanding of our environment. The sight of a crackling fire is made more vivid by the sound of its popping embers and the warmth on your skin. These integrated experiences are far greater than the sum of their individual sensory parts. This intricate dance of sensation and perception is not an isolated process. It is deeply interwoven with our memories, our emotions, and our capacity for learning. The limbic system, that ancient core responsible for emotion and memory, constantly interacts with our sensory pathways. A particular scent might instantly transport you back to a childhood memory, not just because of the raw chemical input, but because of the powerful emotional and mnemonic associations linked to it. Our past experiences literally shape how we perceive the present, creating a unique and personal lens through which each of us views the world. So, as we reflect on this journey through our senses, we begin to appreciate that our perception of reality is an exquisite dynamic construct. It is the nervous system's brilliant solution to the challenge of interpreting a vast, complex external world. From the simple transduction of energy into electrical signals, to the complex cortical processing and multimodal integration, our brain tirelessly builds the internal universe we inhabit. But perceiving the world is only half the story. Once we have built this internal model, once we have understood or felt the world around us, we often feel compelled to act. We reach out, we speak, we move. This brings us to the equally astonishing realm of how our nervous system translates intention into action, how it orchestrates the intricate ballet of muscle contractions that allow us to interact with the world we have just perceived. And that, my curious companion, is a journey we shall embark upon next, as we explore the masterful motor systems that bring our inner world into outward expression. For now, let us simply marvel at the profound wonder of how, from mere ripples of energy, our brain paints the vibrant canvas of our perceived reality. If the journey through sensation and perception in our last chapter invited us to ponder how the universe paints itself upon the canvas of our mind, how we construct the very reality we inhabit, then this chapter turns our gaze outward. It is here, in the intricate dance of our motor systems, that we discover how our inner world, our intentions, desires, and the rich tapestry of our perceptions translates into tangible action, shaping the world around us. We move from the quiet act of witnessing to the profound power of doing. Think for a moment about the simplest movement, reaching for a cup of tea, shifting in your seat, or even the subtle adjustment of your gaze as you read these words. Each of these actions, seemingly effortless, is the culmination of an astonishingly complex orchestration within your nervous system. It's a testament to the seamless integration of thought, sensation, and the precise command of countless muscle fibers. How does the spark of an intention, an abstract thought like, quote, 31, become the graceful, coordinated ballet of muscles and bones? This is the grand question we gently explore now, embarking on the adventure of movement. Our capacity for movement, from the most delicate tremor to the most powerful leap, is a testament to the nervous system's executive function. It's the ultimate expression of our agency, the means by which we interact with, modify, and explore our environment. It's how we manifest our will, communicate our emotions, and fulfill our deepest needs. Let us begin at the genesis of voluntary movement, within the vast, silent chambers of the cerebral cortex. Before a single muscle fiber contracts, before a neuron sends its command down the spinal highway, there's a period of intricate planning. This isn't a simple switch flicking on. It's a sophisticated negotiation, a subtle deliberation that occurs primarily in the association areas of our frontal and parietal lobes. The prefrontal cortex, that pinnacle of our executive functions, plays a crucial role here, formulating the overall goal, weighing consequences, and initiating the idea of movement. It asks, what do I want to achieve? Meanwhile, the posterior parietal cortex works in concert, integrating sensory information about our body's current position in space, our, quote, 30°, and the spatial relationship of objects around us. It answers, quote, 38, quote. Together, these regions construct a mental blueprint, a detailed action plan before the actual execution begins. Once the plan is meticulously drafted, it's passed on to the brain's specialized, quote, 39 located in the frontal lobe. This region isn't a single entity, but a complex of interconnected areas, each playing a distinct role in refining and initiating the movement. The primary motor cortex acts as the final command center, sending direct signals to the muscles. It's organized somatotopically, meaning different parts of the body are represented in specific locations, forming a sort of neural map, often depicted as the motor homunculus, a distorted figure where body parts with finer motor control, like hands and face, occupy a larger cortical area. Adjacent to the primary motor cortex are the premotor cortex and the supplementary motor area. These regions are vital for preparing and sequencing movements. The premotor cortex is particularly active when we plan movements based on external cues. For instance, reaching to catch a ball we see approaching. The supplementary motor area, on the other hand, is more involved in internally generated movements, those we initiate from memory or intention, such as playing a musical instrument or performing a learned routine. It helps sequence complex movements, ensuring one action flows smoothly into the next. Together, these cortical areas don't just tell muscles to contract, they orchestrate a symphony of contractions and relaxations, ensuring fluidity and purpose. But the brain's motor system is far more than just a direct command line from cortex to muscle. It involves an exquisite network of subcortical structures that refine, coordinate, and smooth our movements, making them graceful rather than jerky, purposeful rather than clumsy. Two such structures stand out: the basal ganglia and the cerebellum. Let's first consider the Sus-46, a collection of interconnected nuclei deep within the cerebrum. These structures act like a sophisticated gatekeeper, or perhaps a master editor for movement. They don't initiate movement directly, but rather they play a critical role in selecting and enabling desired movements, while inhibiting unwanted ones. Imagine a vast library of potential movements. The basal ganglia help you choose the right book and keep the others quietly on the shelf. They are crucial for initiating self-generated movements, for learning habits, and for the smooth, fluid execution of learned motor sequences. When the basal ganglia falter, as in conditions like Parkinson's disease, we witness the profound impact: difficulty initiating movement, tremors, and rigidity, a stark illustration of their vital role in the effortless flow of action. Then there is the quote 47 or quote 48, nestled at the back of our heads, posterior to the brainstem. This remarkable structure is a master coordinator and a silent guardian of balance and precision. It receives a continuous stream of sensory information from our muscles, joints, and inner ear, informing it about our body's current position and movement. Simultaneously, it receives copies of the motor commands being sent from the cortex. The cerebellum compares these two streams of information, what the brain intended to do versus what the body is actually doing, and then sends corrective signals back to the motor cortex and brainstem. It's like a highly skilled quality control manager, constantly fine-tuning our movements, ensuring accuracy, timing and smooth execution. Have you ever marveled at the grace of a dancer, the precision of a surgeon, or the effortless balance of a tightrope walker? Much of that finesse is attributable to the cerebellum. It is also a critical player in quote 49, allowing us to refine skills through practice, transforming clumsy attempts into fluid expertise. Think of learning to ride a bicycle or play a musical instrument. The cerebellum is diligently at work, calibrating and recalibrating, helping your movements become automatic and unconscious. From the motor cortex, the commands for voluntary movement descend through powerful neural pathways, primarily the quote 50, which travels down through the brain stem and into the spinal cord. This tract acts as the primary conduit for voluntary motor commands, connecting the brain's executive centers directly to the motor neurons in the spinal cord. These motor neurons are the final common pathway, the last stop before the muscles. They extend their axons out of the spinal cord, directly innervating the muscle fibers and triggering their contraction. It's a direct, elegant chain of command from thought to fiber. But not all movement is voluntary, consciously willed. Our nervous system also orchestrates a vast array of involuntary movements and reflexes, rapid, automatic responses that are essential for our survival and daily functioning. Think of withdrawing your hand instantly from a hot stove, or the knee-jerk reflex when a doctor taps your patella tendon. These reflexes often bypass the higher brain centers, being processed directly within the spinal cord itself. This allows for incredibly fast reactions, minimizing potential harm before the conscious brain even fully registers the event. The brainstem also plays a crucial role in involuntary motor control, particularly in regulating posture, balance, and basic locomotion. It integrates sensory information from our eyes, inner ear, vestibular system, and proprioceptors to maintain our equilibrium against gravity, allowing us to stand, walk, and sit without constantly thinking about it. These deeper, older parts of the brain provide the foundational stability upon which our more complex voluntary movements are built. What ties all of this together, creating a truly dynamic and adaptive motor system, is the constant feedback loop between our sensory and motor systems. Movement is not a one-way street. As we initiate and execute a movement, a continuous stream of proprioceptive information, data about the position and movement of our limbs and body, flows back to the brain. Specialized proprioceptors in our muscles, tendons, and joints constantly report their status. How stretched a muscle is, how much tension is in a tendon, what angle a joint is at. This sensory feedback is crucial for the cerebellum and motor cortex to make real-time adjustments, ensuring accuracy and smoothness. Without it, our movements would be clumsy, uncoordinated, and utterly lacking in grace. Imagine trying to walk in the dark on an uneven surface, without any sensation in your feet. It would be a perilous, almost impossible task. This continuous sensory-motor integration is what allows us to adapt to changing environments, learn new skills, and execute complex motor sequences with such effortless precision. It's a conversation, a constant dialogue between what we intend to do, what we are doing, and what the world demands of us. So, when we consider the simple act of reaching for that cup of tea, we are no longer seeing a mere physical action. We are witnessing the culmination of an internal dialogue, an intention sparked in the prefrontal cortex, a spatial map formed in the parietal lobe, a movement plan refined by the premotor and supplementary motor areas, a command issued by the primary motor cortex, edited and smoothed by the basal ganglia, coordinated and balanced by the cerebellum, transmitted down the spinal cord, and executed by precise muscle contractions, all under the watchful eye of continuous sensory feedback. This journey from thought to action is, in its essence, the story of how our inner self reaches out to touch the external world. It is the mechanism through which we express our creativity, build our civilizations, nurture our relationships, and explore the very limits of our physical capabilities. Our movements are not merely mechanical, they are imbued with meaning, intent, and the unique signature of our individual being. They are the physical manifestation of our consciousness, allowing us to sculpt our reality, one deliberate, graceful, or even clumsy action at a time. This incredible capacity for movement reminds us once more of the profound wonder embedded within the quiet architecture of our nervous system. Welcome back, my dear fellow explorer. As the evening deepens and the world outside settles into its quiet rhythm, let us turn our gaze inward once more, for our journey through the nervous system, while having illuminated so much, still holds its most profound mysteries. We have traced the evolutionary whispers from single-celled stirrings to the magnificent symphony of the human brain, dissecting its components, understanding its electrical dance, and marvelling at its intricate architecture. We've seen the neurons fire, the glial cells support, the systems coordinate, and the grand processes of perception and motor control unfold. Yet, as we stand at the precipice of our understanding, looking out over the vast shimmering landscape of what we do know, a new vista opens before us, the boundless expanse of what remains unknown, the questions that continue to whisper at the edges of our scientific consciousness. These are not merely gaps in data, but profound philosophical chasms that challenge our very definitions of self, reality, and existence. Perhaps the most enigmatic of these, the ultimate frontier, is the mystery of consciousness itself. We've touched upon it, acknowledged its emergent quality. But how does the ceaseless, intricate ballet of cells and chemicals within your skull give rise to you, to the vibrant, subjective tapestry of your inner world, to the feeling of the cool air on your skin, the taste of a sweet berry, the ache of a memory, or the sudden flash of an idea? Consciousness is not a singular, neatly packaged entity residing in one corner of the brain, waiting to be discovered under a microscope. Instead, it seems to be an emergent property, a grand crescendo born from the integrated activity of countless neural networks, humming in harmonious complexity across the entire system. It's the conductor of the orchestra, yes, but also the music, the musicians, and the very air within the concert hall all at once. It's the feeling of awareness, the ability to reflect on your own thoughts and feelings, to know that you know, to be self-aware. This subjective experience, this what it is like to be, is often referred to as qualia, the raw, irreducible sensory and emotional qualities of experience. How does the firing of a specific set of neurons translate into the redness of a rose or the melancholic strain of a cello? This, my friend, is the hard problem of consciousness, a question that continues to humble even the most brilliant minds. The brain, in its infinite wisdom, doesn't merely receive the world, it actively constructs it. We've learned that your perception isn't a passive recording, but a dynamic creative act. Think of it as a master storyteller, constantly weaving a coherent narrative from fragmented sensory inputs, coloured by your memories, expectations and emotional state. The light waves hitting your retina, the pressure waves vibrating your eardrum, these are just raw data. It's your brain that transforms them into the vivid, three-dimensional world you inhabit, complete with sounds, smells, and textures. Consider optical illusions, those playful tricks of light and shadow that make you question what you see. They aren't failures of your vision. They are exquisite demonstrations of your brain's relentless drive to make sense, to impose order and meaning where there might be none. It fills in the gaps, predicts what should be there based on past experience, and often makes educated guesses. Your brain is a magnificent prediction machine, constantly generating hypotheses about the world and then testing them against incoming sensory information. This means that your reality, my reality, indeed, everyone's reality, is profoundly unique, filtered through the lens of individual history and neural architecture. What you perceive as real is in essence a highly sophisticated, personalized simulation, crafted moment by moment within the quiet theatre of your mind. And what of the self? This sense of I that persists through time, this continuous thread of identity that anchors your experiences. Where does it reside within this bustling neural metropolis? Is it a CEO in the frontal lobe, issuing commands, or a quiet observer watching the show? The truth, as always, is far more intricate and beautiful. Your sense of self is not a fixed entity, but a dynamic, ongoing narrative, constantly being written and rewritten by the interplay of memory, emotion, and current experience. Every memory you recall, every decision you make, every emotion you feel contributes to this evolving story. Your brain is a master autobiographer, constantly integrating your past with your present, projecting possibilities into your future. It is the sum of your experiences, the unique pattern of synaptic connections forged over a lifetime of learning and living. And this self is remarkably adaptable, capable of growth, change, and even profound transformation. The very plasticity of your nervous system, its ability to rewire and reorganize itself, underscores the fluid nature of who you are. Speaking of memory, let us delve deeper into its labyrinthine corridors. We've learned that the formation of memories relies on the remarkable phenomenon of synaptic plasticity, the strengthening or weakening of connections between neurons. This microscopic dance, often referred to as long-term potentiation, LTP, or long-term depression, LTD, is the cellular basis of learning. But memory is far more than just a storage facility. It is a creative act, a constant process of reconstruction rather than simple retrieval. When you recall an event, your brain doesn't pull up a perfect, pristine recording. Instead, it reassembles fragments, filling in details, often subtly altering the narrative with each retelling. This is why memories can be so vivid, yet sometimes surprisingly inaccurate, influenced by subsequent events, emotions, and even suggestions. The hippocampus, nestled deep within your temporal lobe, plays a crucial role in forming new declarative memories, memories of facts and events. But once formed, these memories are often consolidated and distributed across various cortical areas, becoming integrated into your broader knowledge network. From the fleeting echoes of short-term memory to the enduring sagas of long-term memory, from the conscious recall of facts, declarative, to the unconscious mastery of skills, procedural. Your nervous system orchestrates an astounding array of mnemonic feats, constantly learning, adapting, and informing your every interaction with the world. Then there are the vibrant hues of emotion and the relentless currents of motivation, the twin forces that propel us through life. We know the limbic system, with its amygdala for fear and pleasure, its hippocampus for memory, and its hypothalamus for regulating basic drives, is central to this. But how do these primal responses intertwine with our rational thoughts, our complex decision-making? Emotions are not mere distractions. They are powerful signals offering vital information about our environment and our internal state. They colour our perceptions, influence our judgments, and often serve as the hidden architects of our choices. The brain's reward system, particularly the intricate dopamine pathways, is a master of motivation. It drives us toward behaviors essential for survival and well-being, seeking food, water, connection, and novel experiences. But this system, so vital for our flourishing, also holds the keys to vulnerability, playing a central role in the development of addiction, where the pursuit of pleasure can override all other considerations. Understanding this delicate balance, the interplay between our ancient emotional core and our sophisticated rational capacities, is crucial to comprehending the full spectrum of human experience. Our journey began with the simplest stirrings of life, and it culminates in the most complex, yet paradoxically, the most adaptable of all structures, the adult human brain. For a long time, it was believed that the brain, once mature, was largely fixed, its circuits set in stone. But modern neuroscience has unveiled a far more dynamic truth: neuroplasticity. Your brain is an ever-changing canvas, constantly rewiring itself in response to new experiences, learning, and even injury. This remarkable capacity for change is what allows you to learn a new language, master a musical instrument, or recover from a stroke. New neural pathways can be formed, existing ones strengthened or pruned, and even the functions of certain brain areas can be reorganized. This ongoing process of adaptation extends throughout life, underscoring the profound potential for growth and transformation that resides within each of us. It is why we can continue to learn, to evolve, and to shape our minds, even into our twilight years. As we bring our peaceful exploration to a close, we must acknowledge that our understanding of the nervous system is still in its nascent stages. The further we delve, the more intricate and awe-inspiring the landscape becomes. What lies on the horizon of discovery? Scientists are pushing the boundaries, developing sophisticated brain-computer interfaces that allow thought to control external devices, offering hope to those with paralysis. We are mapping brain circuits with unprecedented detail, unraveling the complexities of neurodegenerative diseases like Alzheimer's and Parkinson's, and exploring novel therapies. The intersection of artificial intelligence and neuroscience is blossoming, raising fascinating questions about the nature of intelligence, consciousness, and what it means to be alive. The journey through the nervous system is, in essence, a journey into the very heart of existence. It is the universe exploring itself, through the intricate dance of electrical impulses and chemical messengers within your own skull. It is the mechanism by which you perceive beauty, feel love, ponder the cosmos and question your own being. So, as you drift into the quiet contemplation of the night, carry with you this profound sense of wonder. Remember that within you, a silent, intricate ballet of cells is dancing, creating your every thought, every feeling, every breath. It is a testament to billions of years of evolution, a masterpiece of biological engineering, and the most extraordinary mystery of all. May this knowledge deepen your appreciation for the magnificent, integrated whole that is you, and ignite a lifelong curiosity for the wonders that still await discovery, both within and without.